Hi everybody. In today's video on 9.1 nervous system, I would like to talk about part E. That is to compare impulse transmission along the axon and across the synapse. But before I can do that, I need to explain the functions of synapse. Let's look at the first function of a synapse. Synapse is necessary to transmit impulse between adjacent neurons. Adjacent being the neurons that are side by side. But then one wonders why there must be a synaptic cleft to transmit this impulse. Wouldn't it be just easier if the two synaptic knob and dendrite were attached directly? Okay, so I'm going to go on to the second function which will help to explain to you why there must be a synaptic cleft. Let me use your situation as an example to explain synapse second function. While you are watching this video, you are receiving stimulus about new biology knowledge on nervous system. At the same time, you may be receiving stimulus from your stomach because you are hungry, or stimulus from your ears because you just heard the notification bell on your handphone, or maybe even stimulus from your body telling you you are tired and you need to go to sleep. Now, imagine if the nerves that are transmitting impulse for each one of these stimulus have no synapse. Then the nerve will directly transmit the impulse from the sensory organs straight to the effectors, meaning that you are going to respond to all four stimulus at the same time. Can you imagine trying to study, eat, and scroll through your handphone while sleeping? Now that's impossible, and even if you are going to do it, it's going to stress you out. So luckily, we don't have that problem because the synapse can actually help to filter less important stimuli. So then you wonder, how does the synapse know which is an important stimuli and which is not? So this is where the synapse or the synaptic cleft becomes important. You see, if the nerves are directly connected, then electrical impulse will be transmitted by saltatory conduction or localized current. But because of the synaptic cleft, you find that the electrical impulse will have to be converted into a chemical impulse, and that chemical impulse will slowly form EPSP on the adjacent dendrite. Now, only if the EPSP reaches threshold will there be an action potential that will form a new electrical impulse that will be transmitted once again. So, if the EPSP does not reach threshold, then that impulse will stop in this neuron. So that is why, depending on how much EPSP is present in each of the nerves, we find we end up focusing on only one stimuli at a time. So in this example, let's say your tiredness, your hunger, as well as your urge to look at the handphone is unable to make EPSP reach threshold, then you will not go to your bed, you will not go to the kitchen, nor will you pick up your handphone. Instead, you find that the nerve that is transmitting impulse about biology is able to accumulate EPSP to reach threshold. So automatically action potential is generated and you continue to focus on your studies. So this way, only important impulses get transmitted. The third function of the synapse is as an integrating center. From the previous example, you notice that we are receiving a lot of stimulus. So the synapse will actually combine whatever impulse from the stimulus and determine the different pathways for the transmission of impulse. From the picture, I'm sure you can predict the fourth function of the synapse. That is, as a switch. The synapse can actually switch on and switch off the transmission of an impulse. The synapse that can switch on the transmission of an impulse is called an excitatory synapse, and the one that switches off the transmission of an impulse is called an inhibitory synapse. The excitatory synapse actually can receive the impulse in two different ways. One is by converging, that is combining all the impulse that it receives, and the other is by diverge, that is receiving the impulse and then passing the impulse to many different pathways. So let me explain each one. I have two examples of how the excitatory synapse does convergent pathways. For my first example, let's say the boy who was standing quietly just now faces a dog that is barking. Now the boy is not running yet, although he looks a bit stressed, is because the bark of the dog is not able to cause the EPSP to reach threshold. So when the dog barks the first time or even the second time, since the EPSP does not reach threshold, action potential is not generated and the boy does not run. But if I change the situation where now the dog is barking continuously, the boy is going to run. And if we check his membrane potential, what has happened is you find that the impulse that is continuously happening causes an accumulative effect. Over here, there is two impulses, but because the two impulses are far away, they do not cause the membrane potential to reach threshold. But when the impulse is one after another, then we find the membrane potential or the EPSP accumulates until it reaches threshold, and then automatically action potential is generated. To explain this situation further, I'm going to use this model of the synapse which I introduced to you in my video on synapse. Of course, this is a more simplified version. If you want to understand the detailed process of impulse transmission across the synapse, please view my video on synapse. Here I am going to explain in brief. Now, the first situation when the boy did not run was because he received a weak stimulus that only caused a few vesicles to move to the presynaptic membrane. So, only a little bit of neurotransmitter diffused across the synaptic cleft and caused only a few sodium voltage gated channels to open. So when there was an influx of sodium, EPSP formed, but not enough to reach action potential, and that is why he did not run. Now let's take the second situation. That is when the dog started barking fiercely. Now he was going to receive the same stimulus from the same neuron, but the stimulus is going to be released in rapid succession. That means one after another. So when that happens, you see, more vesicles will move to the presynaptic membrane, more neurotransmitters will diffuse across the synaptic cleft, more sodium voltage gated channels will open, more sodium influx will occur, and more EPSP will form. What happens next is, because this happens one after the other almost immediately, the EPSP now will converge. That means they will combine and the amount will be high enough to reach threshold potential and action potential. So a new electrical impulse can be generated, and this is what causes the boy to start to run. So this method of the EPSP becoming TP and then AP is also known as temporal summation or facilitative effect. Okay, remember this is due to the same stimulus being released in rapid succession. Next, I show you a different situation. This time, the boy is not only hearing the barking of the dog, but he is seeing the dog running towards him. So now there are two different stimulus. This will make the boy run. So if we look at his membrane potential, what has happened is the two different stimulus. One is the sound of the dog barking, and the other is the sight of the dog chasing him, has caused EPSP to reach threshold potential and automatically achieve action potential. Let me explain further. In the case where now there are two different stimulus passed through two different neurons, but the condition is both of the stimulus are released simultaneously at the same time. So here I have one stimulus from the ear and one stimulus from the eye. Because they arrive at the same time, they cause the vesicles to move down at the same time. So a lot of neurotransmitters diffuse across the synaptic cleft and cause EPSP to form. But because this is happening at the same time, the EPSP can converge to form threshold potential and action potential. So a new stimulus is formed and causes the boy to run. This form of excitatory synapse involves what we call as spatial summation, or also called an additive effect. So the additive effect is when one stimulus added with another stimulus being released at the same time. So please make sure you are clear with the difference between temporal summation and spatial summation, or facilitative effect and additive effect. So I've talked about the excitatory synapse involving the converging process. Let's move on to the divergent process. Now I'm going to talk about excitatory synapse, but using the concept of divergent. The same situation, the dog is barking, and for the boy, this can be either temporal summation or spatial summation, but when he wants to respond, he has a few options. He is not only going to run, he is also going to look for a stone to throw at the dog, and he's also going to look for a tree to climb to safety. So you see the stimulus is one, but the response is many. This is only possible with a synapse, because if you have neurons that are connected without a synapse, then he can only do one activity, that is run. But because of the presence of a synapse, he can do a few activities using only one stimulus. This is known as divergent. Okay, the impulse comes from one neuron. For example, seeing the dog barking or chasing him. So that impulse then will be transmitted to many other neurons. That is the neuron to the leg, okay, to make him run. The neurons to the hand to make him pick up a stone. Or the neurons to both his hands and legs to make him climb. All this is only possible because of the presence of synapse. Now, when we talk about the function of a synapse as a switch, remember it can also be an inhibitory synapse. So once again, I take the example of the dog barking and the boy. Now, in some cases, instead of running, some people go into a state of panic where they are unable to do anything. They know they are in danger, but they can't move. Okay, this is because the inhibitory synapse has been stimulated. You see in this diagram, you can see there is a neuron transmitting impulse from, let's say, his eyes because he can see the dog, and another neuron transmitting impulse from his ears because he can hear the dog. But you see there is a third impulse coming in. This is known as an inhibitory impulse. So, although you see the EPSP is like going up and almost reaching threshold, but because of this inhibitory impulse, it brings down the EPSP, preventing any action potential from being generated. Let me explain further. The inhibitory synapse is a little different compared to the excitatory synapse. The difference is that over here in the excitatory synapse, you have sodium voltage gated channel. But for an inhibitory synapse, it will be replaced with potassium voltage gated channel. And then in the dendrites, instead of experiencing an influx of sodium, the dendrites will be rich in potassium. So what happens is, when a neurotransmitter arrives, it will cause the potassium gated channel to open. And this will cause an efflux of potassium ions. So when the potassium ions diffuse out, a process of repolarization and hyperpolarization occurs. That means now the membrane potential in this neuron will become more negative. So that is known as IPSP, inhibitory postsynaptic potential. So once there is an IPSP, where the membrane potential is now extremely negative, there can be no generation of an action potential. So that is why the person panics and is unable to respond. Now talk about synapse final function. To do that, can you analyze this diagram and try to identify what is most likely wrong with this picture? If you notice, these yellow circles are neurotransmitters, and they show as if the neurotransmitters are moving from the synaptic knob to the dendrite and from the dendrite to the synaptic knob. Now this is where it's wrong. You see, the synapse can only transmit impulse in one direction. The reason for this is because of the location of the synaptic vesicles. You can only find synaptic vesicles in synaptic knobs. And the receptor proteins are only found on the dendrite. So that is why the neurotransmitters must always move from the synaptic knob to the dendrite. So now we have come to the end of this video. With the information from this video added to the information from the video on transmission of impulse along the axon, as well as the video on synapse, you should be able to compare the mechanisms of impulse transmission along both axon and synapse. So until I meet you in my next video, goodbye.